Hello, I'm Christopher Barnes,
And I'm Rwik,
and we will be presenting to you the incredible and exciting frontier of epigenetics.
Epigenetics is the study and exploration of all those processes which lead to changes in our gene expression without any changes in our underlying genomic sequences.
That's right. And the word epigenetics derives from the Greek roots epi, meaning upon, and genesis, meaning origin. The term, coined in 1942 by an esteemed scientist from Great Britain by the name of Conrad Hall Waddington.
Okay, so we know that epigenetics is all these changes to our gene expression, but where exactly are these changes happening?
Well, in short, these changes occur inside of the nucleus of eukaryotic cells, our cells, where DNA exists in condensed packaging around proteins, creating these fundamental units in chromatin called nucleosomes. And epigenetic factors include entities which dynamically alter these chromatin structures to either be more open and accessible to transcription factors, and therefore increase gene expression, or causing them to be more condensed and inaccessible to transcription factors, leading to a condition called gene silencing. So these changes can happen within our cells and in turn directly affect us.
Yes, but not only us. Hence, the crucial importance of epigenetics studies, because it not only helps regulate development, immunity, health, disease of everyone here now, but also those that will be coming in the future.
Now, our review presents various kinds of epigenetic modifications at levels of chromatin, DNA, RNA, and proteins in the context of transgenerational inheritance, the immune system, and diseases.
Histone proteins that are part of the chromatin structure contain N-terminal tails comprised of amino acid residues, which are modified by enzymes leading to epigenetic modifications. In addition, major epigenetic roles are also exerted by DNA methylation, non-coding RNA, and prions.
Which can collectively lead to health conditions like cancer and other diseases, which affect development, neurodegeneration, immunity, metabolism, and aging. And over the years, research has proven that epigenetics has a very significant role in almost all aspects of health, disease, and immunity. So it's very important to develop cutting-edge technology to study the world of epigenetics.
Absolutely. And one of the most important and popular tools in this field is chip sequencing, or ChIP-seq, which maps genome-wide localization of targeted proteins of interest. The earliest publications on ChIP are from Dr. David Gilmore and Dr. John Liss from Cornell University in the 1980s, and the method has evolved with increasing popularity over the years. The protocol starts with cells and tissue which are cross-linked to immobilize protein-chromatin interactions, sonicated to obtain workable sizes of chromatin, and then followed by immunoprecipitation with antibodies of interest. The immunoprecipitated DNA is purified for further analysis, as very nicely presented here in this image from a recent publication by Nakato and Sakata.
We can also map genome-wide regions of both open and closed chromatin using a procedure called ATAC-seq, which was first reported in 2013 in a very high-impact publication from Stanford University. In ATAC-seq, sequencing adapters, indicated here in red and blue, are loaded onto an enzyme transposase, which allows for taggmentation, a process which hosts simultaneous fragmenting and tagging of targeted genomic locations with sequencing adapters. These adapters are inserted only in regions of open chromatin and can generate high-throughput sequencing library fragments that can later be amplified by PCR reactions. The reason why ATAC-seq is so great is because in its entirety, ATAC-seq requires much less starting material and preparation time, which gives it an advantage to other like-purposed methods.
But there are so many additional methods that can be used in epigenetics research, as shown here.
That's exactly right. For example, there's cut and tag, which answers similar questions as ChIP, Hi-C, which shows chromosome conformation, and RNA-seq, which reveals transcriptomes. But that doesn't stop there. There are many more other methods of epigenetic exploration, and if you're interested in learning about these techniques and implementing them in your research, then Active Motif can help, offering over 2 decades of expertise in the field.
In addition to techniques, however, we also need equipment for processing samples to enable high-throughput research methodology.
And that's exactly right. An active Motif can also help you there.
This is because one such equipment is Pixel, which is published in Nucleic Acids Research in 2019 from the University of Washington and Active Motif. Using Pixel, you can rapidly sonicate up to 96 samples with high consistency for a variety of epigenetics-based studies.
And the number of applications for this machine are growing day by day. Which is great because though we've started gaining ground in the field of epigenetics, there is much more of interesting world to be explored. And if we've engaged your interest, please feel free to email us or connect with us through Active Motif's many social media platforms.
We would like to thank MDPI, the scholarly encyclopedia community, SciPlayer, and Active Motif for this wonderful opportunity.
And most of all, we'd like to thank you for watching. We hope you've enjoyed this video. Signing off for now.
I'm Rwik.
I'm Christopher Barnes.
And we look forward to hearing from you.